Hello students, today I will give a lecture on prokaryotic and eukaryotic cells. Let us start with the introduction. There are two major types of cells, prokaryotic and eukaryotic, based on their complexity. The general characteristic features of the two groups are: first, let us take up the characteristics of prokaryotic cells. Prokaryotic cells are found in bacteria, archaea and cyanobacteria or blue-green algae, and eukaryotic cells include all the other cells such as protista, fungal, plant and animal cells. The structures of a typical prokaryotic cell are described in more detail here. First, let us take up the common cell morphologies and arrangements. Cell morphologies commonly found in prokaryotic cells are coccus, plural cocci or round in shape, bacillus, plural bacilli or rod shape, vibrio, plural vibrios or curved rods, coccobacillus or plural coccobacilli or short rod, spirillum in plural spirilla or spiral, spirochete, plural spirochetes or long, loose, helical spiral, etc. In addition to cellular shape, prokaryotic cells may group together in certain arrangements like coccus, plural cocci or single coccus, diplococcus, plural diplococci or pair of two cocci, tetrad, plural tetrads or grouping of four cells arranged in a square, streptococcus, plural streptococci or chain of cocci, staphylococcus, plural staphylococci or cluster of cocci, bacillus, plural bacilli or single rod, streptobacillus, plural streptobacilli or a chain of rods. Next, let us see the structures external to the cell wall. The first structure is glycocalyx. The glycocalyx, also called capsule or slime layer or extracellular polysaccharide, is a gelatinous polysaccharide and or polypeptide covering lying externally to the cell wall. The exact chemical composition varies depending on the species. Capsules may protect pathogens from phagocytosis and enable adherence to surfaces, prevent desiccation and may provide nutrients. The next structure is flagella. Flagella are long filamentous appendages consisting of a filament, hook and vessel body and help to push the cell by rotating. The filament is composed of the globular protein flagellin, which is arranged in several intertwined chains that form a helix around a hollow core. Motile bacteria move by taxis in two ways: positive taxis or movement towards an attractant and negative taxis or movement away from a repellent. The next are the axial filaments. Axial filaments are similar to flagella except that they wrap around the cell, and spiral cells that move by means of an axial filament or endoflagellum are called spirochetes. Next are the fimbriae and pili. Fimbriae and pili are short, thin appendages. Fimbriae are usually many and help the cells adhere to surfaces, while pili are only one or two per cells and help join the cells during conjugation for the transfer of DNA and are called sex pili, or are used for special types of movement like twitching in Pseudomonas aeruginosa, Neisseria gonorrhoeae and some strains of E. coli, or the gliding movement of Myxobacteria. The next structure is the cell wall. Prokaryotic cell walls are composed of peptidoglycan in bacteria or pseudopeptidoglycan in archaea. Cell wall in gram-positive bacteria are characterized by a thick peptidoglycan layer, whereas in gram-negative bacterial cells, the cell walls are characterized by a thin peptidoglycan layer surrounded by an outer membrane. Next, let us see the structures internal to the cell wall. The first structure is the plasma or cytoplasmic membrane. The bacterial plasma membrane is a phospholipid bilayer with peripheral and integral proteins, while the archaeal membranes are composed of fatty acids that are ether-linked to phospholipids. The plasma membrane of some prokaryotes contains enfoldings called chromatophores that contain pigments involved in photosynthesis. The plasma membrane may also form irregular enfoldings called mesosomes that function either in DNA replication and cell division or excretion of exoenzymes. The plasma membrane encloses the cytoplasm and is selectively permeable. Movement across the membrane may be by passive processes like diffusion and osmosis or by active transport. The next structure is the cytoplasm. Cytoplasm is the fluid component of the cell surrounded by the plasma membrane. The cytoplasm is composed mostly of water along with inorganic and organic molecules, DNA, ribosomes and inclusions. The next structure is the nucleus. Prokaryotic DNA and DNA-associated proteins are not bound by a complex nuclear membrane and occurs as the nuclear region in the cytoplasm of the cell. Prokaryotic chromosomes are typically circular and haploid or unpaired. In bacteria, nucleate-associated proteins or NAPs interact with the prokaryotic DNA and function like histones of eukaryotic cells, assisting in the organization and packaging of the chromosome. However, in archaea, either NAPs or histone-like DNA organizing proteins associate with the chromosomal DNA to organize the nucleus. The next are the plasmids. Prokaryotic cells may also contain small circular double-stranded extrachromosomal DNA molecules called plasmids. Plasmids are generally many in number per cell and often carry genes that confer advantageous traits such as antibiotic resistance. The next structure is the ribosomes. The cytoplasm of a prokaryotic cell contains numerous ribosomes of the type 70S. Ribosomes are made up of ribosomal RNA and proteins and serve as the site for protein synthesis. The next are the inclusions. Inclusions are reserve deposits found in the cells, and some examples of prokaryotic cell inclusions found in bacteria are metachromatic granules or inorganic phosphate, polysaccharide granules usually of glycogen or starch, lipid inclusions, sulfur granules, carboxysomes containing ribulose 1,5-diphosphate carboxylase, magnetosomes including magnetite or Fe3O4 and gas vacuoles. The next structure are the endospores. Endospores are resting structures formed by some bacteria like Bacillus and Clostridium during unfavorable environmental conditions. The process of endospore formation is called sporulation, and the endospore returns to its vegetative state by a process called germination. The next feature is sociability. Although prokaryotes are unicellular, most of them can form stable aggregated communities in a polymer matrix called biofilms. Cells in biofilms often show distinct patterns of gene expression or phenotypic differentiation in time and space. Bacterial biofilms are more resistant to antibiotics compared to the single bacterial cells and are difficult to remove from the surfaces of their colonization. The next is social environment. Prokaryotes can inhabit nearly all environments. Some archaea and bacteria can even thrive in extreme conditions such as high temperatures called thermophiles or high salinity called halophiles, and such organisms are referred to as extremophiles. Some prokaryotes live in the bodies of other organisms as symbiotic prokaryotes, while many archaea grow as planktons in the oceans. The next is reproduction. Bacteria and archaea reproduce through an asexual mode of reproduction known as binary fission. During binary fission, The genomic DNA undergoes replication followed by division site selection, invagination of the cell envelope and synthesis of new cell wall, resulting in the division of the original cell into two identical parts. Next, let us take up eukaryotes. Eukaryotes include all species of large complex organisms including animals, plants and fungi, and most species of protist microorganisms. Eukaryotes are differentiated from the prokaryotes mainly by their genome enclosed in the nucleus surrounded by the nuclear membrane and the presence of other membrane-bound organelles. Compared to prokaryotes, eukaryotes represent a tiny minority of all living things, and eukaryotic cells generally have a smaller surface area to volume ratio than prokaryotes and thus have lower metabolic rates and longer generation times, except in some cells specialized for metabolism such as the intestinal villi. The main characteristics of eukaryotic cells are: first, let us see the cell features. Eukaryotic cells are larger than prokaryotic cells and vary greatly in their morphology. They have a variety of internal membranes and structures called organelles and a cytoskeleton composed of microtubules, microfilaments and intermediate filaments which help in maintaining the cell's organization and shape. The next is the cell wall and glycocalyx. Cells of fungi, algae and plants and some protists have a cell wall, whereas cells of animals and some protozoans have a sticky extracellular matrix or glycocalyx. The cell walls of plants, many algae and some fungi are made up of cellulose, while most fungal cell wall contain chitin. The next structure is the plasma membrane. The plasma membrane of eukaryotic cells is a phospholipid bilayer containing proteins like the prokaryotic membrane. However, eukaryotic membranes differ from prokaryotes in containing carbohydrates attached to the proteins and sterols as well as glycoproteins and glycolipids. Movement of materials across the plasma membrane occurs by passive and active processes as in prokaryotes. However, in addition to active processes, eukaryotic cell membranes can take materials into the cell via endocytosis, which includes phagocytosis and pinocytosis, or expel matter from the cell via exocytosis. The next are the flagella and cilia. Eukaryotic cells may contain flagella and cilia for locomotion, feeding, or movement of extracellular particles. The next structure is the cytoplasm. The cytoplasm of eukaryotic cells includes everything inside the plasma membrane and excluding the nucleus. Chemically, the cytoplasm of eukaryotic cells resemble those of the prokaryotic cells. However, eukaryotic cytoplasm has a cytoskeleton and exhibits cytoplasmic streaming. The next structure are the ribosomes. Eukaryotic cells contain two types of ribosomes. ATS ribosomes are present in the rough endoplasmic reticulum as membrane-bound ribosomes and in the cytoplasm as free ribosomes, while the 70S ribosomes are present in mitochondria and chloroplasts. The next structure is the nucleus. The nucleus contains DNA in the form of chromosomes surrounded by a nuclear envelope which is connected to a system of membrane in the cytoplasm called the endoplasmic reticulum. The nucleolus is located inside the nucleus of eukaryotic cells and serve as the site of ribosomal synthesis and the site for the first stages of ribosome assembly. The next structure is the cytoskeleton. Cytoskeletons are composed of microfilaments, intermediate filaments and microtubules. They provide structural support in eukaryotic cells and serve as a network for transport of intracellular materials. The next feature is the presence of centrosomes. The centrosome consists of the pericentriolar area and centrioles. These are microtubule organizing centers that take part in the formation of the spindle apparatus during cell division. Next, let us take up the organelles. Eukaryotic cells contain a variety of specialized membrane-bound structures called the endomembrane system or organelles. The organelles present in eukaryotic cells are vesicles, the endoplasmic reticulum, mitochondria, plastids, and the Golgi apparatus. Next, let us see reproduction in eukaryotes. Sexual reproduction in most eukaryotes involves an alternation between haploid and diploid generations occurring through nuclear fusion or syngamy and meiosis. Nuclear division is often coordinated with cell division and takes place by mitosis. The next feature is cell association or hierarchy. In the plant and animal kingdom, cells associate to form tissues, tissues to organs, organs to organ system, which finally makes the whole organism. Thus, the major differences between the prokaryotic and eukaryotic cells may be summarized as follows. First, the size of prokaryotic cells range from 0.2 to 5 micrometer in diameter, while that of eukaryotic cells range from 10 to 40 micrometer in diameter. Secondly, prokaryotic cells do not have a nuclear membrane, and the nuclear material consisting of a single chromosome lies in the cytoplasm and is called nucleus. Eukaryotic cells, on the other hand, have a distinct nucleus bound by a nuclear envelope. The third feature is prokaryotic cells lack membrane-bound organelles, while eukaryotic cells contain membrane-bound organelles like lysosomes, Golgi complex, endoplasmic reticulum, mitochondria, chloroplast, etc. in their cytoplasm. The next feature is flagella in prokaryotes consists of two protein building blocks, while flagella, if present in eukaryotes, are complex consisting of multiple microtubules. The next feature is prokaryotes possesses glycocalyx as a capsule or slime layer, while it is present only in some cells that lack a cell wall in eukaryotes. The sixth feature is cell wall is usually present in prokaryotes and is chemically complex. Example, peptidoglycan of typical bacterial cell wall. Eukaryotes lack cell wall commonly and, if present, are chemically simple. The seventh feature is the plasma membrane in prokaryotes do not contain carbohydrates and generally lack sterols, while that of eukaryotes contain carbohydrates and have sterols that serve as receptors. The cytoplasm in prokaryotes does not contain cytoskeleton or cytoplasmic streaming, while it is present in eukaryotic cells. Prokaryotic cells contain smaller ribosomes or 70S type, while eukaryotic cells contain small or 70S ribosomes in organelles and larger or 80S ribosomes in the cytoplasm. The DNA in prokaryotes is arranged as a single circular chromosome and lacks histone proteins, while it is present as multiple linear chromosomes associated with histones in eukaryotes. The next is cell division in prokaryotes is generally by binary fission, while it occurs by mitosis in eukaryotes. Next, sexual reproduction in prokaryotes does not involve meiosis and DNA transfer occurs through conjugation, while sexual reproduction in eukaryotes involves meiosis. Let us now see the conclusion. Thus, we have seen that the prokaryotes are mostly unicellular cells with simpler cellular organization, and they live in nearly all environments including extreme conditions, while the eukaryotic cells are mostly multicellular and much more complex compared to those of the prokaryotes. Unlike the prokaryotes, the eukaryotic cells contain many subcellular structures that play important roles in energy balance, metabolism, and gene expression. Thank you.